I am Cheryl Abrigo with USF Technology Transfer Tech Tours. Today we are here in the lab of Dr. Hana Totary-Jain, who we're visiting with today. She is the Associate Professor of Molecular Pharmacology and Physiology. Thank you for taking time to come talk to us today. Recently, you just had an article published in Cell Host and Microbe in June. Congratulations. Can you please tell us a bit about your research that led to this article?
Yes, in fact, this article was not within my expertise because we were investigating cardiovascular disease, and in particular, we were looking into benign tumor of infants, which is characterized with blood vessel growth. And we wanted to do small RNA-seq, and we found that there is one of the largest human or primate-specific microRNA cluster highly expressed in these tumors. And it is characterized also, it's found only usually in the placenta. So we were trying to investigate what's the role of this microRNA cluster. And we did do a CRISPR to activate this gene, and we find that it's induced interferon. As you know, interferon should be only induced upon viral infection. It's never on its own. And we started to think what is going on here. And from the literature in the placenta, it has been shown that trophoblast cells in the placenta constitutively all the time induce interferon type 3. And no one knows what's the reason. And here we found that, here we know this is this gene. And they also showed that it is a microRNA cluster. It is 46 pre-microRNA genes. And they also showed that this microRNA do not induce interferon. So we wanted to look more deep into this sequence, and we found that within this 46 microRNA, pre-microRNA, there is also 365 alloretrotransposons, which they form double-strand RNA. They cause viral mimicry response, and that's why they induce interferon. So we did very simple experiment and to differentiate between the microRNA and these alloretrotransposons. And we used dicer knockout cells, which we eliminated all the microRNA, and we ended up with only the allus. And we see that they do indeed induce interferon. And that was our breakthrough here, because we really discovered the way the placenta protects against viral infection. Because the placenta all the time express this gene, and this gene makes it fools it. It makes the placenta feel that there is all the time viral infection and produce all the time interferon. So they are proactive, and they are all the time protecting the baby from. And in fact, most of the viruses, they do not transmit to the fetus, because this mechanism is what protects the babies.
That is incredible.
Yeah.
That's incredible. So with our office, you have an impressive 6 granted patents, and you have 11 disclosures in prosecution. Can you tell us about some of your other areas of interest that bring out these?
Yes. So regarding this project, we immediately put a patent to use the alloretrotransposon as a vehicle to protect from viral infection, not only in the placenta, but it can also help other infectious diseases in adults as well. And we can also target allus because there is a lot of disease which are caused from over-inflammation or over-infection, over-reaction, or like sterile inflammation. So we can target allu to reduce this sterile inflammation, because that's what is happening in the placenta. The allus are inducing sterile inflammation. Before that, my lab started with trying to develop first of its kind cell-selective therapy to treat atherosclerosis or cardiovascular disease. And we did develop one of its kind microRNA switch. We could target the plaque-forming cells, like smooth muscle cell, inflammatory cells, infiltrated inflammatory cells, while at the same time we could protect the endothelium. because endothelium are very important cells which protect from thrombus formation. So most of the therapies which are available today, including the drug-eluting stents, are not selective. They inhibit the smooth muscle cell, other things, but not, they also inhibit endothelial cells. And we have several nice papers which we show that we could selectively inhibit neointimal formation while restoring, and we have also other patents which we also put to accelerate vessel healing. So yeah, that's one of we are also using the same technique, the microRNA switch, to even use it to do gene editing in a cell selective therapy. So we can use this tool in a variety of things. So we have patents on all this.
And how would you like these to go to the next level? What would be your goal for these?
I would love to see some more funding. We have already one grant, and I'm trying to apply for more grants, but it would be nice if we can have a partnership with some companies, some sponsor research or licensing, because obviously we cannot work on all these technologies at the same time. And yeah, I would like to have a little bit more funding so we can hire more postdocs, more students, give this opportunity to more students to learn, and prepare also the next generation to carry on this work, because it's very important. It's part of our mission.
Right, and that brings me to my final question of how important is additional funding. And I know you mentioned the students, and I know you have a high school student here, which is very exciting. I think that's a great opportunity.
Yeah.
So
yeah.
It would just be great.
Yeah. So exactly, we need more support so we can really make more impact and help patients at the end, because that's one of my missions, is to be able to bring it to the patients. And I think we have a great opportunity and great approach to solve many diseases, or at least help find treatment for many diseases.
Thank you so much for sharing this with us. It's been fascinating, especially the article that was published. So interesting. Thank you for joining us today with our Tech Tour series, and I look forward to seeing you again next time.